Hello and welcome to today's webinar, A Question of Life or Death, Differentiating Between Healthy and Apoptotic Cells. Your host will be Dr. Judith Langenig, one of our antibody product managers here at Bio-Rad ABD Ceratec. Judith received both her BSc and PhD degrees in molecular biology from the University of Dundee, Scotland. Her PhD thesis research focused on elucidating STAT signaling pathways in the social amoeba Dictyostelium discoideum. For her postdoctoral research, Judith moved to the MRC Laboratory of Molecular Biology in Cambridge, where she investigated the role of the Fanconi anemia DNA repair pathway. Since leaving academic research, Judith has held product management positions in both Germany and the United Kingdom. As we're trying to make this webinar as interactive as possible, please submit questions or comments via the Q&A box located at the bottom of the screen throughout the webinar. We will try to answer as many questions as possible in the session following the actual presentation. Should any questions remain unanswered, we will mail you a reply. The webinar will be recorded and an on-demand version will be available shortly after the webinar on the Bio-Rad ABD Ceratec website. Should you encounter any technical problems during the webinar, please click on the question mark at the bottom of the screen. This will bring up a technical guide which describes solutions for the most common technical problems. I hope you enjoyed the webinar, and I now hand over to Judith.
Hello and welcome everybody. So what topics will we be covering in today's webinar? There will be a quick introduction into the definition of the term apoptosis and some historic information on apoptosis research. From there, we will be moving on to the different apoptosis pathways, the intrinsic versus the extrinsic apoptosis pathway. We will also be discussing the differences between apoptosis and necrosis. This section will be followed by reviewing the different stages of apoptosis and the ultrastructures, morphologies that define these stages. We will then be reviewing the different assay and methodology types that can be used to determine if a cell is apoptotic. These assays are also helpful to determine where exactly in the apoptosis pathway a cell is located. From there, we will be moving on to the tips and tricks for apoptosis assay selection part of the webinar. Finally, the webinar will conclude with a Q&A section. What does the word apoptosis mean, and what is the origin of the word? Apoptosis is a Greek noun generated from the prefix apo and ptosis, and is generally translated as falling off. The term is commonly associated with the imagery of leaves falling from trees or flower petals falling off a flower. It has a very autumnal theme and has an element of separation. If you are interested in learning more about the term and its origin, George Eduardo Duque Parra has written a historical note called Note on the Origin and History of the Term Apoptosis that has been published in the Anatomical Record in 2005. The term apoptosis was first introduced by Kerr et al. In the 1972 paper Apoptosis, a basic biological phenomenon with wide-ranging implications in tissue kinetics. The paper is now considered a classic and has been cited over 15,000 times. Although apoptosis is a commonly used term to describe programmed cell death, it is important to note that apoptosis is not the only form of programmed cell death. Unfortunately, due to time constraints, I will not have the time to delve deeper into the topic of the different programmed cell death mechanisms during this webinar. Much of what we know about apoptosis today originates from research performed on model organisms such as the nematode C. elegans. During development into an adult nematode, 1,090 somatic cells are generated, of which exactly 131 die through apoptosis at dedicated developmental stages. This work and other related discoveries in C. elegans have been so valuable to elucidate the mechanisms of apoptosis that Sidney Brenner, Robert Horwitz, and John Sulston were awarded the Nobel Prize for Physiology or Medicine for their work in 2002. Why is apoptosis research important, and what is the link to disease states? From what we discussed on the previous slide about C. elegans development, you will recall that apoptosis is critical for embryogenesis and development. Another well-known example for the role of apoptosis in development is the formation of human hands. These start out as paddle-like structures in which fingers emerge due to apoptosis. Aberrant and dysregulated apoptosis plays a role in many diseases. Indeed, the invasion of apoptosis has been identified by Hanahan and Weinberg as one of the 10 hallmarks of cancer. Please be aware that depending on the research field, this hallmark might be somewhat contested. Aberrant apoptosis also plays a key role in the context of infectious diseases. Most notably, certain viruses have developed mechanisms to inhibit specific proteins in the apoptosis machinery. In the case of neurodegenerative diseases, such as Alzheimer's disease or Parkinson's disease, increased apoptosis rates can be observed. Having provided a brief introduction into apoptosis, I would like to discuss the two major apoptosis pathways. These are the extrinsic and intrinsic apoptosis pathways. The extrinsic pathway, shown on the left-hand side of the diagram, is mediated through death receptors. One example of how this pathway can be activated is through binding of the Fas ligand to the Fas receptor, which in humans is also known as cluster of differentiation 95 or CD95. The intrinsic pathway, on the other hand, shown in the middle of the diagram, is not mediated through receptors, but through mitochondria in response to apoptotic stimuli such as DNA damage. These stimuli trigger intracellular signaling cascades. Please note that crosstalk between the two pathways exist as highlighted by the green box in the diagram. Changes in survival factor levels, such as loss of growth factors or cytokines, also contribute to the regulation of apoptosis signaling pathways. These contributions are shown on the right-hand side of the diagram. With regards to the intrinsic apoptosis pathway, the signaling cascade starts and is mediated by BCL2 homology proteins. Members of this protein family can either act as apoptosis inducers or inhibitors by forming homo or heterodimers. In the case of the classical intrinsic apoptosis pathway, the two main BCL2 homology proteins are BAX, which stands for BCL2-associated X protein, and BAC, which stands for BCL2 antagonist or killer. These critical proteins are marked in the diagram with the numbers 1 and 2. Upon activation by interaction with the pro-apoptotic factor BID, which stands for BH3 interacting domain death agonist, BAC and BAX are inserted into the outer mitochondrial membrane as homo or heterodimers. This process is highlighted in the diagram by the number 3 and results in mitochondrial outer membrane permeabilization or MOMP. Mitochondrial outer membrane permeabilization is regarded as the point of no return in apoptosis. Until reaching this point, cells can still be repaired and survive. However, upon the onset of MOMP, a cell is committed to undergo apoptosis. MOMP ultimately results in the formation of a mitochondrial apoptosis-induced channel, which is a large pore that facilitates the release of pro-apoptotic factors such as cytochrome C into the cytoplasm. This release is marked by the number 4 in the diagram. Upon its release to the cytoplasm, cytochrome C interacts with the adaptor protein APAF, which stands for apoptotic protease activating factor 1. The nucleotide deoxy-ATP, or DATP, binds to APAF to mediate its activation. APAF then recruits procaspase 9, which collectively with ATP and cytochrome C forms a complex called the apoptosome. The apoptosome has a very unique structure and is marked in the diagram as number 5. Formation of the apoptosome triggers cleavage of procaspase 9 into caspase 9, which is the active form of the caspase enzyme. This step is marked by the number 6 in the diagram. It is important to highlight that the word caspase stands for cysteine aspartic protease. Members of the caspase protein family not only have the ability to cleave other caspases, but also a variety of non-caspase substrates, such as the caspase-activated DNase CAD. Caspase 9 is an initiator caspase, which triggers activation of the caspase cascade by cleaving procaspase 3 and procaspase 7 into their active forms. These two caspases are effector caspases, and their cleavage is marked in the diagram as step 8. We will discuss the role of caspases and their activation in more detail in the apoptosis stages and related assay section of the webinar. For more detailed information about the caspase cascade and the intrinsic apoptosis pathway, please refer to www.abdserotech.com/caspases. The extrinsic apoptosis pathway is initiated by the binding of ligands to death receptors, shown in the diagram as number 1. Upon ligand binding, the receptors dimerize or oligomerize, resulting in the activation and recruitment of adapter proteins such as FAD, which stands for Fas-associated protein with death domain. The FAD recruitment is shown by number 2 in the diagram and enables the recruitment of procaspase 8. This step is marked by the number 3 in the diagram. To be a bit more precise, the recruitment of procaspase 8 results in the formation of the death-inducing signaling complex, or DISC, marked by the number 4 in the diagram. Procaspase 8 first oligomerizes and then becomes cleaved into caspase 8. This step is marked by the number 5 in the diagram. Caspase 8 then subsequently cleaves and activates caspase 3. This process is shown as number 6 in the diagram. In some cells, however, extrinsic activation of caspase 8 can also lead to activation of the intrinsic pathway. This pathway crosstalk is mediated through BID. For more detailed information about the extrinsic apoptosis pathway, please refer to www.abdserotech.com/caspases. Now that we have gained a better understanding of apoptosis and the different pathways, I would like to introduce the term necrosis and how necrosis differs from apoptosis. Like the word apoptosis, the term necrosis originates from Greek. The word can be translated as dead or death. In contrast to apoptosis, which is a form of programmed cell death, necrosis is an unprogrammed cell death process occurring in response to external damage or injury. During apoptosis, cells shrink, while during necrosis, cells swell up. Apoptotic cells become engulfed by phagocytes, while in the case of necrosis, cell debris is released. Apoptosis is also a non-inflammatory, non-immunogenic process, while necrosis is inflammatory and immunogenic. However, as we become more aware of new forms of cell death, the described criteria and features to distinguish apoptosis from necrosis become less and less well defined. An example of these newish forms of cell death is necroptosis, which is a programmed form of necrosis that proceeds without the involvement of caspases. Also, the notion that apoptosis is non-immunogenic is an oversimplification, as immunogenic apoptosis or immunogenic cell death has been reported. This phenomenon has been mainly observed in response to chemotherapeutic agents in cancer cells. Distinguishing between the different cell death forms can be tricky, and therefore several methods and time points have to be analyzed in order to confirm the exact death mechanism. The variety and selection of assays for the detection of the different apoptosis stages will be discussed in the next section. If you have any questions about the webinar so far, questions related to your apoptosis experiments, or comments, please feel free to submit these via the Q&A box located at the bottom of your screen. For those of you who joined us later, the webinar recording will be made available after the webinar on the Bio-Rad ABD Serotech website. Apoptosis is undertaken in very distinct stages that are characterized by certain morphological features and hallmarks. This slide gives an overview of the main stages, but please note that depending on the literature, the order and names of the stages might be a bit different. Once initiated, apoptosis results in the activation of cell signaling cascades. These cascades, which are mediated by members of the caspase protein family, lead to cell shrinkage and cells rounding up. The shrinking stage is followed by organelle changes and membrane blebbing. Blebbing signals that a cell cytoskeleton has been rearranged, or rather been detached from the plasma membrane. Blebs may be observed for a short period of time as cellular protrusions. The onset of blebbing is followed by nuclear condensation, also known as pycnosis. Pycnosis is followed by DNA fragmentation or karyorrhexis. One of the final stages of apoptosis is the formation of apoptotic bodies, which become phagocytosed by macrophages. Apoptotic bodies, for example, are very distinct cellular structures that can be observed by microscopy. In general, every apoptosis stage shows very distinct morphological or ultrastructural features. One way to determine and analyze a specific apoptosis stage is through live cell imaging. When this technique is combined with time-lapse movies, the entire apoptosis process can be observed from onset to cell death. This method is particularly useful for the visualization of blebs. Electron microscopy is a classical and high-resolution method to observe apoptosis and traditionally used for tissue analysis. Tissues can also be stained with dyes such as hematoxylin and eosin, which indeed can be used to visualize macrophages that have engulfed apoptotic cells. Antibody-based methods such as immunohistochemistry and immunocytochemistry are also very popular. Tissues and cells tend to be stained with antibodies that have been raised against key apoptotic proteins such as cytochrome C, APOF1, and caspases. The nucleic acid binding dye acridin orange is also routinely used to visualize engulfed apoptotic cells or apoptotic bodies. When bound to single-stranded nucleic acids such as mRNA, the dye emits red light, while binding to double-stranded DNA results in the emission of green light. Due to this feature, the dye is often used for the analysis of cell cycle stages and for the analysis of cell cycle progression. When present in lysosomes or other low pH compartments, acridin orange upon excitement of blue light fluoresces orange. This feature is used to stain and visualize apoptotic cells present in the lysosomal compartments of macrophages or other phagocytes. In order to analyze apoptosis, three types of methods or assays are commonly used. The first method is the analysis of changes in the mitochondrial membrane potential. If you remember the intrinsic apoptosis pathway, you might recall the crucial role that mitochondria play in this pathway and the changes happening to the mitochondrial membrane. Indeed, these changes result in a reduction of the mitochondrial membrane potential, highlighted in green in the diagram. The mitochondrial membrane potential can be measured with the help of specific dyes in flow cytometry or immunofluorescence experiments. Another readout or analysis tool for early apoptosis is caspase activation, which can either be assessed by flicker, fluorescent labeled inhibitor of caspase assays, or by Western blot analysis. Using antibodies specific for procaspases and active caspase proteins allows distinguishing between inactive and active caspase forms. Molecular weight differences between the proteins can also be observed, which allow a differentiation between healthy and apoptotic cells. Multiplex immunoassays such as Bio-Rad's Bioplex Pro RBM apoptosis multiplex assay can also be used to measure several key apoptosis markers and parameters simultaneously. A decrease in the mitochondrial membrane potential is a sign of increased membrane permeability and hence a sign that a cell has passed the point of no return in terms of committing to apoptosis. Three dyes are commonly used to analyze changes to the mitochondrial membrane potential. These are TMRE, which stands for tetramethylrhodamine ethyl ester, TMRM, which stands for tetramethylrhodamine methyl ester, and JC1. The mitochondrial membrane potential MitoPTM, TMRE, and TMRM kits shown in the left-hand and middle images utilize a change in the fluorescence intensity of the TMRM and TMRE reagents to distinguish healthy from apoptotic cells. TMRM and TMRE dyes aggregate in the mitochondria of non-apoptotic cells and fluoresce bright orange or red. In apoptotic cells, on the other hand, the dyes diffuse throughout the cell. Once dispersed, the reagents assume a monomeric form and display reduced orange-red fluorescence levels. This reduced intensity can be observed in the images labeled dyeing. Not only is the staining intensity reduced, some cells show no staining at all. The JC1 reagent used in the kit shown on the right-hand image easily penetrates into cells and enters the mitochondria. It aggregates in the mitochondria of non-apoptotic cells and fluoresces red, whilst in apoptotic cells it diffuses throughout the cell. Once dispersed, the reagent assumes a monomeric form and fluoresces green. This allows an easy distinction between non-apoptotic red fluorescing cells and apoptotic green fluorescing cells. If you look closely at the right-hand image, you will notice that the cells located at the top are healthy. In contrast to that, the cells shown at the bottom are nearly completely green and therefore can be classed as apoptotic. Earlier in the webinar, we reviewed the caspase cascade and introduced the term initiator and effector caspases. Caspases 3, 6, and 7 are effector caspases, while caspases 2, 8, 9, and 10 are initiator caspases. In order to evaluate if the caspase cascade has been activated, it is important to determine if key procaspases, such as procaspase 9, have been cleaved. This can be done by Western blot analysis with the help of antibodies specific for the active and inactive caspase forms. An example of such an experiment is shown here on the left. The experiment investigates activation of human caspase 9 in untreated versus apoptotic HeLa cells. Apoptosis was induced with the apoptosis-inducing agent staurosporin. In lane 1, a band of around 30 kilodalton can be observed in the treated HeLa lysate, while no band can be detected in the untreated lysate. Lanes 3 and 4 have been probed with an antibody recognizing the inactive caspase 9 protein. You will notice the higher molecular weight of the inactive or procaspase 9 protein. According to UniProt, the molecular weight of the human procaspase 9 protein is 43 kilodalton, while the active cleaved caspase has a molecular weight of 30 kilodalton. In addition to antibodies recognizing active initiator caspases, we also offer a variety of antibodies for the detection of active effector caspases, such as active anticaspase 3 and anticaspase 7 antibodies. Caspase activity can also be measured by utilizing fluorophore-labeled inhibitors of caspases, or short flicker molecules. These inhibitors are specific for activated caspases and are comprised of 3 parts. The first part is the fluorophore, which can either be FAM, which stands for carboxyfluorescent and is a green-emitting dye, or SR, which stands for sulforhodamine and is a red-emitting fluorophore. The second part is the specific caspase inhibitor sequence, which is followed by fluoromethylketone, or FMK. An example of the cell-permeable caspase 9 flicker molecule is shown here. The inhibitor is labeled to FAM and contains the caspase inhibitor sequence LIHD. As caspases tend to bind sequences comprising 4 amino acids, the inhibitors are designed to resemble a typical caspase cleavage motif. Indeed, the inhibitors are competing with actual substrates for caspase binding. However, once bound, the inhibitor binds covalently and irreversibly to the caspase. This is mediated by the FMK moiety. Unbound inhibitors either diffuse or are removed during the WASP procedure stipulated in the protocol. Depending on whether the inhibitor was labeled with SR or FAM, apoptotic cells either fluoresce green or red. This fluorescence can either be measured by flow cytometry, immunofluorescence, or by reading the absorbance on a plate reader. In the image shown here, you can see 2 apoptotic cells. The cell on the left-hand side is greener and therefore shows more caspase activity compared to the right-hand cell. Earlier on in the webinar, we discussed plebbing and the reattachment of the cytoskeleton from the plasma membrane. The plasma membrane in general provides a key readout of apoptotic changes, as in the early stages of apoptosis, the plasma membrane is rearranged, resulting in changes to the cell membrane's phospholipid asymmetry. The effects of these rearrangements are the display of phosphatidylserine, or PS, molecules shown here in purple in the graphic at the outer plasma membrane. In healthy cells, PS is exclusively located at the inner membrane, and therefore the exposure of PS is an indication of early apoptosis. This feature or characteristic of apoptotic cells is utilized in annexin 5 assays. The anticoagulant annexin 5 has a high binding affinity to PS in the presence of calcium, and the binding of labeled annexin 5 can therefore be used to determine if PS has been exposed to the outer plasma membrane. Binding of annexin 5 to PS is therefore indicative of a rearranged plasma membrane structure and ultimately apoptosis. Annexin 5 assays are performed by flow cytometry utilizing fluorophore-labeled annexin 5. One of the most common annexin 5 labels is FitsAll. Most annexin 5 kits include the Viability dye propylium iodide, which enables the distinction between dead or secondary necrotic and apoptotic cells. A typical annexin 5 experiment is shown here on the right-hand side, which allows the differentiation of three distinct cell populations: viable, apoptotic, and dead or secondary necrotic cells. Several time points tend to be analyzed in order to observe a shift in the cellular populations and to determine the effect of treatments. For more information about the mode of action of annexin, please go to www.abdserotek.com/annexin-5. In the previous slide, we discussed plasma membrane rearrangements upon the onset of apoptosis. However, it is important to highlight that PS molecules have the ability to flip back from the outer to the inner membrane, and that plasma membrane changes are reversible up to a point. This reversibility is shown in image B by the gray double arrow. The purple PS molecules indeed flip back from the outer to the inner plasma membrane. Using annexin 5 assays, one is unable to visualize these reversible and other kinetic apoptosis events. For this type of analysis, another assay type is required, which allows the visualization of apoptosis events in real time by live cell imaging. This assay utilizes a biosensor called PSEVA, which stands for Polarity Sensitive Indicator of Viability and Apoptosis. The biosensor is labeled to a green-emitting dye called IANBD and binds to PS in the presence of calcium. However, only when the probe is bound to PS will the biosensor fluoresce. Unbound PSEVA will not emit a signal, and once PS molecules flip from the outer back to the inner membrane, the PSEVA probe will fall off and stop emitting green light. The PSEVA probe can therefore be compared to a light switch that can be easily switched on and off. As discussed for annexin 5 assays, in order to distinguish early from late apoptotic and dead cells, propylium iodide staining should be performed. The plasma membrane of late apoptotic cells is permeable, allowing propylium iodide to enter these cells and to become intercalated into DNA. Late apoptotic cells will therefore fluoresce red, while early apoptotic cells will fluoresce green. For more information about the PSEVA mode of action, please go to www.abdserotek.com/PSEVA. This assay type is especially useful when comparing knockout to wild-type cell lines, as the assay allows the easy quantification of differences between apoptosis rates. For example, wild-type cells could repair DNA damage and survive, while the mutant cells can't repair the damage and undergo apoptosis. The PSEVA probe is included in the PSEVA Real-Time Apoptosis and Fluorescent Microscopy Kit, which has the product code APO004. In addition to the probe, the kit also includes a vial of propylium iodide. In contrast to annexin 5 kits, no incubation or washing steps are required with the PSEVA kit. You can simply add the probe and propylium iodide and start imaging, provided your medium contains sufficient amounts of calcium. The detailed staining protocol can be found on www.abdserotek.com/PSEVA. An example of a PSEVA experiment is shown below. HeLa cells were either treated with the apoptosis-inducing agent staurosporin or with the vehicle/solvent in which the staurosporin had been dissolved in. Cells were incubated and images were taken at 0, 60, 120 and 270 minutes. Staurosporin-treated cells displayed green fluorescence after 60 minutes. This is indicative of the cell having started to undergo apoptosis and signals that the PSEVA biosensor has bound to PS. Over time, the green fluorescence gets replaced by red fluorescence, a sign of late apoptosis and indicative that propylium iodide has been able to enter the cell. The PSEVA data shown on the previous slide were taken on the Zoe fluorescent cell imager that allows you to image cells on your bench, as the light sheet permits fluorescence imaging in ambient light. Imaging is highly simplified, and the intuitive touchscreen interface allows you to view cells, capture images, and create multi-channel merges. The imager has a flexible operation characterized by a bright field and 3 fluorescence channels. This makes the imager ideal for routine cell culture and more sophisticated imaging applications. Find out more about the Zoe fluorescent cell imager at www.biorad.com/Zoe. When reviewing the different apoptosis stages earlier in the webinar, DNA fragmentation was identified as one of the later stages of apoptosis, which is followed by the formation of apoptotic bodies and phagocytosis. DNA fragmentation is commonly analyzed by two methods. The first method is based on running extracted genomic DNA on agarose gels to determine DNA laddering. The laddering is due to the genomic DNA becoming cleaved by endonucleases into oligosomes of a molecular weight of around 180 base pairs and multiples of these 180 base pairs. The second methodology for determining DNA fragmentation are tunnel assays. The term tunnel stands for terminal deoxynucleotidyl transferase DOTP nick and labeling. As mentioned, endonucleases or DNases such as caspase-activated DNase cut become activated by caspases and cleave DNA into oligonucleosomes. This cleavage results in DNA breaks or nicks, which display a hydroxyl group at the 3' end of the DNA fragment. In a tunnel assay, the enzyme TDT is used to add promylated deoxyuridine triphosphate nucleotides to the hydroxyl group. Once incorporated, the BRDU-DPs can be detected with a biotinylated or fluorophore-labeled anti-BRDU antibody. For more information about tunnel assays, please go to www.abdserotech.com/tunnel. Tunnel assays can be performed by flow cytometry or immunohistochemistry/immunofluorescence. Examples of readily available tunnel assays include the ApoBRDU kits APO001 and APO002. In addition to the critical components identified on the previous slide as the TDT enzyme, BRDU-TPs, and the labeled anti-BRDU antibody, both kits include positive controls. In the case of the flow kit, these controls are frozen positive and negative cells. The positive cells can be used for comparison to your own data. In the case of the IHC kit, two control slides are provided, in addition to the nuclear counters then methyl green and proteinase K to perform proteolytic-induced antigen retrieval. For more information about these tunnel assay kits, please visit our product pages for APO001 and APO002 or go to www.abdserotech.com/tunnel. When describing the mode of action of the Nexin 5 and PCEVA assays, I mentioned that it is important to combine these assays with cell viability dye stainings, such as performing propylium iodide staining. This is especially critical in order to differentiate apoptotic from necrotic or dead cells. There's a variety of dyes on the market covering the fluorescent spectrum. If you would like to learn more about our cell viability dye offering, please go to www.abdserotech.com/cell-health to view our range of ready-drop and VIVA-fix dyes. Since we have reviewed a variety of assays and apoptosis mechanism information, I would like to move on to the more practical part of the webinar by sharing some hands-on tips and tricks with you. Please note that this is the last part of the webinar. Therefore, if you have not already submitted your questions, please feel free to do so via the Q&A box located at the bottom of your screen. I really can't emphasize this next point enough. Control your experiments. Controls are essential to prove that your experiment has worked and that you have not detected an artifact. Controls are also critical when troubleshooting or optimization is required, as you can identify the exact step of the experiment where things have gone wrong. Therefore, make sure to include both positive and negative controls. These could be untreated cells or commonly and well-studied cell types. As many apoptosis-inducing agents are dissolved in solvents, control for the effects of the solvent by including a vehicle-treated control. Include application-specific controls. For example, in IHC experiments, autofluorescence controls are important, as certain tissue types have a natural fluorescence. This control type is less important in Western blotting, where loading controls are commonly used. I have included an image of our PCEVA booklet, which provides detailed information on how to set up a kinetic apoptosis assay by controlling for both autofluorescent and solvents effects. This experimental overview is also meant to provide a rough guideline on the most common controls. Avoid what I call a one-size-fits-all approach and use the same assay and conditions for all of your apoptosis experiments. Every experiment is different, and every cell line is different. Therefore, scientific literature provides only a rough guideline, and apoptosis induction conditions and assays will need to be optimized for every experiment. Carefully consider what you would like to show. You can use apoptosis stages as rough guidelines. For example, use caspase assays to show apoptosis initiation. Use a different assay type, such as a tunnel assay, to show DNA fragmentation. Carefully consider what you would like to disprove. Use apoptosis stages and cell death modes as guidelines. For example, you might want to show that your cells are apoptotic by determining that they are not necrotic. Another example could be that you would like to show that your cells are early rather than late apoptotic. Perform multiple assays, use different methodologies to confirm these results. Also always include a cell morphology analysis by checking the cells and tissues under the microscope. Analyze multiple targets, investigate several crucial proteins. In the case of a Western blot panel, this could include active and procaspases, bugs, APAF1, cytochrome C, and BIT. The results from these experiments could then be confirmed with a mitochondrial membrane potential measurement or a flicker assay, depending on exactly what you would like to show. As so often in life, timing is everything, and it matters when and after what treatment duration you're performing your experimental analysis. Please also bear in mind that apoptosis is a reversible process and that until the point of no return has been reached, cells can still avoid programmed cell death. Therefore, please ensure that your conditions are optimized and that you have selected an assay type allowing the analysis of reversible and early apoptotic events. If you're comparing a newly generated knockout to a wild-type strain for the first time, a tunnel assay might not be the best starting point. In such a case, a live cell imaging or microscopy-based technique might be more suitable. Also, as apoptosis is a reversible process, please ensure that you perform a time course analysis. Try to include kinetic data and live cell imaging data. If you are not analyzing several time points, you might draw the wrong conclusions and said that your cells were unaffected by a treatment, when indeed they were affected, arrested at the cell cycle stages, but managed to repair the damage prior to reaching the point of no return. Failure to measure several time points might also result in the wrong conclusions. For example, you could stipulate that cells died through necrosis when indeed their cell death was mediated through apoptosis. You might have been simply unable to detect apoptosis as you treated the cells for too long or analyzed them too late. There comes a time point by which most apoptotic cells can be stained with propylium iodide and therefore might be labeled as secondary necrotic. If you remember the PCVA data that I showed earlier, one could observe apoptosis as early as 60 minutes. However, after 4.5 hours, nearly all the cells were dead. As apoptosis is a rapid and transient process, make sure to include appropriate controls such as untreated cells for comparison. Observe the cell morphology. Make sure you quickly analyze the cells under the microscope before performing an apoptosis assay. This will reduce the risk of false positives or negatives and also help you to back up your findings. For example, if you see rounded up or shrunken cells, you can be more confident in your flicker staining or active caspase Western blot results. In order to assist you with your apoptosis experiments, we offer a variety of resources, especially if you're intending to analyze apoptosis by flow cytometry or immunohistochemistry. Our application resources might come in handy. For both of these applications, you can listen to our webinars in our webinar library, download protocols, tips and tricks, or view our reagent offering. All mentioned resources can be downloaded at abdserotech.com/applications. If you have problems locating these or would like us to post you a hard copy, simply let us know by submitting a message via the Q&A box. If you are intending to analyze apoptosis by Western blotting, Bio-Rad offers a complete workflow that includes products for sample preparation, electrophoresis, protein transfer, antibodies for immunodetection, and image acquisition and analysis instrumentation. The cancer research antibody offering includes antibodies for analyzing apoptosis and autophagy, cell proliferation, cell signaling, oncoproteins, and tumor suppressor proteins. The antibodies have been tested in a variety of applications and include highly validated PrecisionApp Western blotting antibodies. Each PrecisionApp product has been carefully selected after comprehensive performance testing in up to 12 different wholesale lysates. PrecisionApp products are characterized by their high specificity, high sensitivity, and batch-to-batch consistency. Every antibody lot has to meet the same strict quality control criteria. For more information, browse the PrecisionApp range at www.abdcertec.com/PrecisionApp. In addition to products, the cancer research resources also include literature pieces such as the snapshots from Cell Press, acute myeloid leukemia, melanoma, and mitochondrial dynamics posters. You can download a free copy of the posters on www.abdcertec.com/cancer or request a free hard copy through the Q&A box or our website. The Cancer Research poster webpage also provides links to antibody reagents that can be used to study AML, melanoma, and mitochondrial dynamics. Immunoprecipitation is another antibody-based method used for apoptosis research. Unfortunately, due to time constraints, we could not cover this method during the webinar. Often, immunoprecipitation is used to pull down procaspases, active caspases, and their interaction partners. In some cases, entire complexes, such as the caspase 8 interacting complex, have been immunoprecipitated. For immunoprecipitation experiments, Bio-Rad offers SureBeads protein A and G magnetic beads, as well as a large selection of antibodies tested in immunoprecipitation. These antibodies are added to the magnetic beads and left to incubate. The beads are then magnetized and unbound antibodies are removed. This is followed by adding the cell lysate containing the target protein to the beads. The lysate and beads are left to incubate, the beads are magnetized, the supernatant is removed, and then the beads are washed. The washing steps are followed by elution in a suitable buffer. Beads are magnetized and the immunoprecipitated proteins are collected. The IP sample is denatured and reduced by boiling in sample buffer containing reducing agents such as DTT or beta-mercaptoethanol. The sample is then run on SDS page, followed by a silver or Comasi staining. Protein bands are cut out from these gels and must spectrometrically analyzed. The aim of this analysis is the identification of new interaction partners or protein complex components. Alternatively to mass spectrometry, Western blotting can be performed. When using standard heavy and light chain secondary antibodies for the detection of IP samples, two bands can always be observed, in addition to the protein or proteins of interest. These are the IgG heavy chain at 50 kilodalton and the IgG light chain at 25 kilodalton. These chains originate from the IgG isotype antibodies conjugated to the IP beads, which become released and denatured during the IP sample preparation. The IgG chains result in problems when your protein of interest, or the protein you are intending to pull down, has the same molecular weight as the IgG heavy and light chains. This is a pretty common phenomenon if your protein of interest is like 50 kilodalton or 25 or 30 kilodalton, as these chains will mask your protein of interest, which will make the detection tricky. Also, new interaction partners and all components of a protein complex may not be detectable, as you might have some proteins hiding under the IgG chains. TidyBlot is an HIP conjugated Western blot detection reagent that can be used as a substitute for secondary antibodies. The reagent specifically binds to native antibodies, so it can distinguish between native and denatured antibodies, and based on this feature, TidyBlot only binds to the primary antibody used for Western blot analysis, rather than to any IgGs released during the IP procedure. TidyBlot therefore mitigates the described interferences of IgG heavy and light chains, and can therefore be useful for your apoptosis-related immunoprecipitation experiments. For more information on TidyBlot, please go to www.abdcertec.com/tidyblot. I would like to let you know that we'll be moving on to the Q&A part of the webinar. Thanks to all of you who have submitted questions and have provided feedback. I have seen that a few of you unfortunately had to leave us already. So just in case you've not already done so, please submit more Q&A questions. But also, if you can't receive an answer during the Q&A session, we'll be getting in touch and we send you a reply by email. You can also alternatively contact us by visiting our website, www.abdserotech.com technical, and you can either contact our technical support directly or find out which office to get in touch with. This could either be our office in Oxford in the United Kingdom, our office in Raleigh in the United States, our office in Puchheim, Germany. Alternatively, you can meet us in person at a conference near you. We will be exhibiting at the American Association for Immunologists in Seattle in May. Our booth number there is 727. For the Q&A session, I'm actually joined by a colleague of mine, Dr. Rachel Preston from the R&D department. Rachel graduated from the University of Glamorgan in Wales with a bachelor's degree in human biology and went on to obtain a Master's of Science degree in bioinformatics from Cardiff University, also in Wales. And Rachel has conducted her PhD research at Cardiff University in the group of Dr. Andrew T, where she characterized the tumor suppression function of folliculin. I will now hand over to Rachel to answer the first question, which I believe is related to tunnel assays. Rachel?
Thank you, Judith. So our first question today is about the disadvantages of tunnel assays and what to be aware of when performing this assay type. So tunnel assays and their interpretation have been heavily debated in the literature, and this has resulted in them being refined over the years. As you can imagine, the biggest pitfall with this type of assay is the fact that all DNA double-strand breaks and nicks become labeled. And these damages are not the exclusive result of the endonuclease-mediated DNA fragmentation and may actually occur during the sample preparation procedure. Therefore, controls such as a sample in which no apoptosis has been induced is essential to evaluate the assay. Also, I would always recommend to include a control in which no TDT enzyme has been added. When you're looking to perform a tunnel assay by IHC detection, it's also highly advisable to consult the literature about the best tissue sample preparation method. Proteinase K-mediated antigen retrieval seems to be the preferred method for paraffin-embedded tissues, and this treatment is often combined with microwaving, which seems to improve the sensitivity of the assay irrespective of the fixative. The other thing to bear in mind is if you're using adherent cells in flow cytometry detection-based tunnel assays, is that apoptotic cells, of course, detach. And therefore, it's important that you collect the medium and the detached cells prior to trypsinization of any adherent cells. You can then spin down the medium or your supernatant, and the collected apoptotic cells can then be reconstituted in an appropriate buffer and pooled with the adherent cell population prior to performing flow cytometry analysis. Failure to collect these detached cells may result in failure to detect all stages of apoptosis. Our second question is it best to use microscopy or flow cytometry-based assay for apoptosis in my experiments? Now, this very much depends upon the context of the experiment and the desired measurement criteria. There are several methods of detecting apoptotic events using fluorescent reagents which lend themselves to analysis by either microscopy or flow cytometry. It's certainly feasible to perform the cell counts on the slides to measure the apoptotic cell numbers. However, microscopic techniques lend themselves better to morphological analysis, and in that it's possible to see the apoptotic marker location in the cell structure, for example, the cell membrane when looking at an exin-5 or the mitochondria when looking at JC1. These kinds of images can have a big impact when you need a positive or negative result. However, for quantifying levels of apoptosis in response to varying treatments or incubation times, I find flow-based protocols can allow the generation of statistical data to support your theory. You can also bear in mind the practicalities of the experiments, including factors such as whether you can easily prepare a single-cell suspension for your flow cytometry, or if your cells are better adhered to a slide. The third question is related to controls for mitochondrial membrane potential assays and what reagents should be used as a positive control. So, with regards to mitochondrial membrane potential assays, carbonyl cyanides and chlorophenol hydrazone, or CCCP, is a commonly used reagent to induce mitochondrial membrane polarization. It can therefore be used as a positive control in order to monitor fluorescence intensity changes and color changes of the TMRE, TMRM, and JC1 dye-based mitochondrial assays. And question 4 is, why can't I do live cell imaging time course experiments with flicker molecules? The thing about flicker molecules is they penetrate cells and bind covalently and irreversibly to caspases. Therefore, once they are added, they sort of promote cell survival as caspases become inhibited and therefore can't cleave their substrates or promote the caspase cascade. And based on this feature, the flicker molecules can only be used to visualize certain time points rather than the entire apoptosis time course. So thank you for those questions. I'm now going to pass you over to Judith for another couple of questions.
Thanks a lot, Rachel. I hope these question and answers have been useful for our audience. The next question is actually related to analyzing apoptosis in embryos or whole mounts. And the question is what assay type we would recommend for this type of analysis. I'm sure this is quite relevant to our audience working with zebrafish or other model organisms. And there is no kind of size-fits-it-all answer, but if you review the literature, a lot of the time if people work with zebrafish embryos or other embryos, they perform aquidine orange staining, and this is the most common assay that can actually be seen in the papers. However, as we discussed with the tunnel assay in the first question and throughout the webinar, every assay has its pitfalls and you have to keep certain kind of aspects of the assay in mind. And with the aquidine orange, that is basically the fact that this dye might also stain lysosomes that contain bacterial DNA or DNA of other microorganisms that have been engulfed by macrophages. So this is just important to keep in mind, and this is also important to keep in mind if you're using aquidine orange for other kind of stainings, let's say tissue stainings, because it's hard to distinguish actually the lysosomes that contain the remnants of the apoptotic cells versus those that contain the remnants of other microorganisms. Based on this kind of pitfall of the aquidine orange, the recommendation would be to not only use the aquidine orange staining on its own, but to combine it with other methods. And when you actually have a look in the literature, quite often this assay is combined with doing tunnel assays or caspase 3 activation assays. So again, make sure to back up your results with those of other assays. Don't just go for one assay and draw a conclusion. So the next question is actually related to looking at morphologies. And we talked a lot about morphology analysis before an assay, and the question is what to do, how to do analysis after or in between an assay. And this question is coming from somebody who's thinking of combining a flow cytometry-based tunnel assay with a morphological analysis. And this is actually quite an interesting point. And Rachel and I are wondering whether you could perform the BRDU staining with the tunnel kit and indeed then split the sample prior to doing the RNase treatment and propydium iodide staining. You could then treat one of the bits and do the PI staining, but perform the flow cytometry analysis. However, from the other half, you could actually prepare cytospin. And that cytospin you could actually simply mount on a cover slip, and this would allow you to do the morphological analysis, and you could then also observe the fluorescence. So this would be a suggestion how you can actually combine the flow-based tunnel assay with also doing a morphological analysis. And we discussed this a lot during the webinar. It's always important to look at your cells throughout the microscope, and this gives you another parameter and another kind of indication of where in the apoptosis process your cells are located. The last question, so unfortunately, as we mentioned, we can't answer all the questions that you submitted, but we'll get in touch by email, is a question related to the JC1 assay, the mitochondrial membrane transition kit assay that's using the JC1 dye. And on one slide, the customer points out that we actually showed healthy cells that show green, red, and yellow stainings. And the suggestion is made that the yellow staining is an overlay of the merged green and the red images. But where does the kind of green originate from? And the thought was that the JC1 emits only the red fluorescence in healthy cells as it's present as a polymer. So there's the question, how can we actually see green fluorescence? So in healthy cells, the JC1 dye is spread throughout cells. However, the cationic dye becomes concentrated, or rather accumulates, in the mitochondria due to its positive charge. And in a cytosolic pH environment, the dye fluorescence is green. However, in polarized mitochondria, the lower pH value causes a fluorescent shift to the red. Due to this, healthy cells appear as an overlay of red and green mitochondrial speckling. So I hope this answers your question and gives you an idea why you can see these three colors. I would like to say thank you for all of you who attended this webinar and who submitted questions. You've been a brilliant and very active audience. For those of you who have not received an answer to your question yet, we will be in touch by email over the next couple of days. May I wish you all the best for your apoptosis experiments and your research in general. Goodbye from Rachel and myself.